So what are enzymes? Enzymes are basically biocatalysts because they are mostly involved in increasing the rate of biological reactions or biochemical reactions. So we can call enzymes as biocatalysts. Initially, scientists thought that all enzymes are proteins, but now it has been identified that some enzymes can also be RNA molecules, and such enzymes, which are basically RNA molecules, are referred to as ribozymes. Earlier, there was a phrase saying all enzymes are proteins, but not all proteins are enzymes. But now they have identified that there are some enzymes in our living systems which are basically nucleotides, which are basically RNA molecules, which is a polynucleotide chain, which can still carry out catalysis of a chemical reaction. So, say you have a chemical reaction where A gives rise to B. A is the substrate of the chemical reaction, B is the product of a chemical reaction. If there is no enzyme, then this reaction is going to take a very long time to proceed. So we need somebody to increase the rate of this chemical reaction, to speeden up, to hasten up this particular reaction, and that's why we have an enzyme. An enzyme mostly exists in its tertiary structure. If you remember, we had learnt about the tertiary structure being like a woolen ball. A protein thread is rolled over itself, is coiled over itself to form a hollow woolen ball like structure. So, if an enzyme has to perform its function normally, it has to exist in its tertiary structure. And when an enzyme coils up into a hollow woolen ball like structure, there will be certain pockets on its surface. So, for example, there's a pocket or a crevice or a depression on its surface, and this depression is what is referred to as an active site, and it is in the active site that the substrate molecule comes and fits into it, and then the enzyme catalyzes its conversion into the product. So because an enzyme exists in a tertiary structure, the surface of the enzyme has lot of depressions, has lot of crevices or pockets. One such crevice or pocket is referred to as the active site, and as you can see, enzymes are the ones which catalyze the reaction through this crevice or a pocket where the substrate has to come and sit. So remember, the substrate comes and binds over there on the active site, and then the enzyme catalyzes the reaction.